Good morning, everybody. Today, we're going to kick off the next panel. Sumant, Kulkarni, and myself will be chairing the session. And as mentioned, this is a panel on advances in treatment of Alzheimer's and cognitive disorders. Just a quick introduction, myself, Murali Gopalakrishnan. I'm with AbbVie, and I head up the research and evaluation efforts in the company as part of the corporate strategy office and based in Chicago. I'll let Sumant say a word about himself, and then we'll move on. Sumant?
I'm going to do a mic drop after this. But my name is Sumant Kulkarni. I'm a managing director with Canaccord Genuity, which is an investment bank. Our franchise was one of the first on Wall Street to cover neuroscience stocks. Thanks to Leonard Sachs and Sachs Associates for having us here. I'd like to say happy neuro year to everyone. And if we believe, Harry, a large percentage of that happiness, especially from an investor perspective, is thanks to my co-moderator Murali here for AbbVie's acquisition of CerovoMed. With that, I'll turn it over to Alexander.
Yeah. We'll do an intro quickly as we'll walk through very quickly. As we do the introduction, we'll also do a mic check. So if you cannot hear, please shout, okay? Please do that. But before we pass it on to everybody and have them introduce, I'm just going to flip the slide over here. You heard from Harry this morning, right? This is an underwhelming area, and we disagree with that, right? It's good to disagree with Harry once in a while. So this is going to be a very overwhelming area. And partly there's a lot of topics to cover here. As schematically shown on this slide, we could spend the next 2 or 3 days here talking about Alzheimer's and other cognitive disorders, but certainly we don't have that much time, and I think we probably have about 45 minutes left now. So what we thought we'd do as a team here is to really focus on a couple of these areas. I bolded a few of them that we'll touch on today. And before we jump into all of that, as Sumant mentioned, we'll just go around the table here and have a quick round of introductions of our esteemed panelist who is going to be providing very valuable insights. So with that, I'll hand it over to Alex. First, you want to go first? And let's do a mic check as well. Make sure we can hear you.
So I hope you can understand me. I've told Jenny I got 4 kids, so maybe I don't need the mic at all. But yeah, Alexander, I'm a German physician scientist, MD, PhD, 30 years in the industry. During one of those career steps, I was also part of the memantine story, which feels like generations ago, but always been in the field of neuroscience. And among other things, I'm also a co-founder and executive chairman of Galimedix.
Okay.
Jenny.
Hello, everyone, and thank you for the invite to join the panel. So I'm Jenny Laird. My role at Lilly is leading our external innovation, so search and evaluation for neuroscience globally. And I'm based in London, but we have a global team, based across the US and other places. And so, again, my background is, like everyone, I guess, neuroscience. But Lilly is a global pharmaceutical company. We've become a little bit more newsworthy, I guess, in the last year or so with all of the obesity news. But just like diabetes and obesity, where Lilly has been in the field for essentially since the 1920s, when we partnered with the University of Toronto to develop insulin, we've also been in the neuroscience space for a really long time. So, Lilly's been working on Alzheimer's for more than 30 years. And it is a difficult and challenging space, but, we certainly really strongly believe that you have to take bold bets in these complex, difficult diseases, and persistence pays off. This is not a field for kind of dabbling, perhaps.
Exactly. John.
Okay.
My name is John Allam. I'm a CEO at CerovoMed. We're a publicly traded company based in Boston on NASDAQ, ticker symbol CRVO. Our lead asset is an oral drug, neflamapimod, which targets the early phase of the neurodegenerative process, synaptic dysfunction, by blocking the effects of neuroinflammation on the synapse, including effects on tau aggregation and the development of tau pathology. Our lead indication now is in dementia with Lewy bodies, where we have positive phase 2A data that's been published in Nature Communications about a year ago, and more recently in Neurology. The Neurology result is about looking at the phase 2A data and looking at the patients with early stage disease ahead of where there's significant neuronal loss versus later on, and that result is actually, there's a rather remarkable increase in the level of efficacy in that early stage population. And it's that group that we're focused in now enrolling into a phase 2b study, which is planned to complete enrollment in the first half of this year and read out by the end of this year.
Thanks, John.
I'll just say that one last sentence. We're not presenting at this conference for a variety of different reasons, but please do look up our website, CerovoMed.com, and there is a presentation this coming Thursday in a different context that has been announced there. And if you want to learn more, please do participate next Thursday.
Thanks, John. Lisa?
Good morning, everybody. My name is Lisa Ricciardi. I'm the President and CEO of Cognition Therapeutics, which is a New York-based neurodegeneration company focused on early, mild to moderate Alzheimer's, like John, dementia with Lewy bodies, as well as at the back of the eye, dry AMD. Our lead program is an oral agent, so we're pretty excited about potentially being a game changer in one or more areas. Our two lead programs are reading out this year, middle of the year, proof of concept in Alzheimer's disease, very exciting, followed by our studies in a mild to moderate, a little bit after John's population, DLB patients. I like to say we cover most of the world together. We'll collaborate by the time this is over.
Got
Murali,
it.
right?
Great. All right. Over to Todd. All right.
Hello. Hi, I'm Todd Haim. I'm at the National Institute on Aging, which is part of the NIH. I'm the NIA Senior Advisor on Biomedical Innovation. And within that role, I direct our seed funding programs, which are non-dilutive SBIR grant funding programs to startups. We actually, this past year, we provided just about $150 million to startups just in 1 year and through this program. Currently have over 30 Alzheimer's disease startups developing therapeutics that are in our portfolio. The nice thing is we are able to take bets and really look at different targets, different types of therapeutics, and fund a wide landscape so that we can really see the whole picture. And I'll actually stop there because I'm really excited for the discussion.
Great. Well, thank you. How did we do the audio? Was it okay? Got it. All right. So as you saw, we have representation on the panel from, I would say, small pharma, big pharma, and the NIH. So it'll be a good discussion today. We're going to start off first with the topic that's pretty familiar to many of you, approaches based on A beta. And we're going to use a couple of slides, not because we want to read through all of this stuff on the slides, but really what we want to do is to kind of go through various points in a meaningful and a logical way so that we don't get stuck with one topic and don't proceed to the other. So I'm going to hand it over to my colleague, my co-chair here, Sumant, who's going to sort of introduce this section, provide an emphasis on what we learned so far, and again, from an investor perspective primarily, what are the implications as we go forward in this space? And we'll sort of cover three buckets as outlined on this slide. Sumant.
Thanks, Murali. So we'll start with amyloid beta. For a long time, amyloid beta was kind of the zombie hypothesis, but it came back in a big way when we had aducanumab, and then we had all the controversy around that. Then we had lecanemab being approved, and now donanemab's waiting in the wings for approval. From an investor perspective, if you looked at stocks of companies trending into the phase 3 data sets, there's typically been a fear of missing out, and those stocks tend to go up a lot. We did not have that happen with Biogen, mainly because I think there was a lot of investor fatigue around the Alzheimer's area, but then we got Eisai approved for lecanemab. So there is still some excitement, I would say, around that. Granted, these are intravenous products. The launches are going to be measured compared to small molecules. But with that, I'll ask Jenny to lead it off, kind of give us a state of the union on amyloid beta, if you would, and then we'll have others chime in on some of the other topics.
I'm definitely not qualified for a State of the Union. I'm sure many people.
After 30 years in Alzheimer's, you are the most qualified person to do that. Let's go there.
Clearly, this is. I guess that it was a zombie hypothesis for others. It wasn't for us. So something that Lilly has worked on a lot over the last few years, and again, it comes back to that what I said in the intro, that it really does help to have learned from past. They were failures, but they're successes in other ways because you learn a great deal about the patient populations. And I think, and the kind of things that are going to work so that at least you can get yourself to a place where you really, truly can test the hypothesis. Because you might say that in the past, we had lots of attempts, but it's not so clear that people were really managing to eliminate A beta from the brain or, to produce a really substantial reduction. Or indeed even test drugs in patients that actually had Alzheimer's, who had amyloid in their brain. So that's been the sort of the biomarkers, which the last panel talked about a lot, and I completely agree. Biomarkers is super important in this field. And I guess one thing I might bring up compared to, to build on what the last panel said, is it's really important to select appropriate patients. I mean, obviously, for Alzheimer's, they need to have both amyloid and tau to actually have Alzheimer's. But also, I think one of the things that we've learned, and I honestly think it's one of the key factors, certainly during the development process, is being able to stage the patients appropriately. Again, several others here are talking about that you don't necessarily, if the neurodegeneration process has gone a long way along and patients are in a later stage, it's much more difficult to expect that you're going to be able to bring back function or see a reduction in the rate of decline if they're already quite declined. Equally, in the context of a clinical trial, if patients are very early on, they may not be declining very quickly. So the placebo group, obviously the two groups have to separate. And if that separation, if that curve is not very steep, it makes it a very long process. It's much more difficult. So the trick, I think, for us was picking the Goldilocks patients in the middle that were declining rapidly, but enough to detect a signal, but that they weren't so far gone that it wasn't going to be possible to observe a preservation of cognition in the same way. So the key is not just picking the right patients, although that's essential. It is also picking the right stage of the disease. And I think that's probably true of others too. The next bit comes commercially, how all of the things we're learning in the clinical trial space, how that translates into the real world to find people who how does all this play out for all the different companies? It's going to be really interesting to see.
Thanks for that, Jenny. So you mentioned biomarkers. Clearly, this panel is about Alzheimer's and cognitive disorders, which are not Alzheimer's. So with that, I'll turn it over to John to discuss the approach there. And for full disclosure purposes, Canaccord was advising one of the sides on your going public. So I'd like to make that disclosure. But with that, John, I'll turn it over to you to talk about your approach, which is biomarker-based.
So I'll actually start by extending a little bit on Jenny's comments about this earlier stage. I think the real opportunity for the industry is that an in-between population that has enough disease that's based on a functional deficit rather than frank neuronal death, and then a later stage which is driven by neuronal death and true neurodegeneration. That middle phase in the field is called the phase of synaptic dysfunction. I think part of what my own view of what donanemab and lecanemab have is they each target very specific synaptotoxic versions of A-beta, which allows them to actually have an effect on synaptic dysfunction, which allows for actually some level of improvement. You see this very clearly in the lecanemab data, where there is actually improvement. And you see it in the donanemab data where you use tau PET scanning to identify that sweet spot in between group that don't have significant neuronal loss but have enough disease and synaptic dysfunction to be able to treat. And when you have reversal, you can see more robust differences from placebo treatment. And I think, Sumant's question to me was around, we've done that effectively the same work in DLB, where we're not using a PET scan to identify where they are in terms of how much synaptic dysfunction versus neuronal loss. We're using a blood-based biomarker, plasma p-tau 181, which actually does correlate quite well both to amyloid markers as well as tau PET scanning in the DLB population. And what we find is when you go into and there's a threshold level. If you're below that threshold, using this blood test, plasma p-tau 181, the level of treatment response in the DLB population with our drug, analyzed as Cohen's d effect size, is twofold higher. And you see effects that are really rather remarkable. And if we can confirm this in phase 2B, we'll be in great shape. But it is, I think, in some sense, I view as 2023 as the year of synaptic dysfunction comes of age, of this concept that's been out in the field, that if you could find this early spot before there's too much neuronal loss, you still have to have disease, but you have treatable disease. I think this is really going to be the path forward for us, and absolutely not having to just depend on imaging, but being able to have plasma correlates of that is that much more opens the path forward.
So we'll take that to the next level. You've said a lot about what's happened and your approaches. So I guess we'll save the tougher part of this for the last. Lisa and then Alex, could you comment on how you're looking about what's going to happen in the future? How do you optimize clinical benefit, maximize that in terms of trials?
Well, I think it's in the first phase, in phase 2B, certainly. At this point, it's about put up your best data that you can. So you really go after your best patients possible. The patients who are further along, we by no means would we say that they're not going to be able to benefit if you have the drug as we do, which seems to be acting on the underlying disease process. But you have to look at it in a completely different time frame. They're not going to be able to show early responses in phase 2. It's more like in AD, you have to go out to looking for disease progression effects. And I think that's, again, the concept could be that you focus on the patients where you can get robust clinical signals, and maybe even go to the market first in that group of patients. And then once you're there, there's a lot more you can do with it. And it's a little bit not different than multiple sclerosis. Relapsing remitting MS is it's a treatable group. It's where there's less neurodegeneration. You can do all your dose finding, you can get results, you can go to the market with that. And then there's opportunity to optimize in relapsing progressive and more progressive, more advanced patient populations.
And the same type of question for Lisa and Alex. How would you opine on all of these topics that we just discussed?
Alex, you want to go?
Yeah, why can't we make up? Maybe I'll start, Lisa. Thanks. Yeah, I think what we also need to do is not only look for cognition as the lead symptom, of course, in Alzheimer's disease, but also look for behavioral changes in the patient's impact on activities of daily living, trying to better understand the patients, and then also characterize our drugs and see in a larger, let's say, phase 2, kind of try to see which type of patients is benefiting the most. Biomarkers, I think that has been mentioned several times, and that is clear. We do need biomarkers, plasma biomarkers, especially that can help us to better understand the progression of the disease, maybe even allow us to forecast disease progression in a single patient. But I fear that we are still far away from that. Our analytical methods have become much better. I still don't see the validated biomarker that we have been looking for the last decades. Maybe we should also re-emphasize focusing on better understanding the patients. What I do expect that will happen in the future is also that on top of the recent progress that we made with the antibody modalities, and please remember that's the first progress we had for 20 years. Memantine in 2002 in US and 4 in Europe was the last new drug that was approved. Now we have 2 and likely 3 new drugs. However, seeing the profile of those drugs, I think unfortunately for the patient, there is still the need for further drugs and maybe, and I'm a strong believer in that, for further modalities. Maybe antibodies are not the only, maybe even not the ideal modality to treat a CNS disease. I'm still a strong believer in small molecules. Let's see what the future brings.
Let's hope there's a small molecule or two in the field.
Get the microphone. Get the microphone, please.
Oh, sorry. So yes, I agree with much of what John and Alexander have said. I was doing some research coming out here, and I found this paper, all these biomarkers for DLB, and I thought, oh my God, how could I not know this? And the list of contributors was 2 full pages. It's every PI we've all worked with. It's incredible. And then you get down to 6 biomarkers and they're like not really linked to disease. And so the point is we're making progress, but it's slow. And I think if you work in Alzheimer's disease, you go, well, we have the canonical biomarkers. Everybody's looking at tau and various forms of tau and amyloid, and ALS brought new biomarkers this year. And so progress is being made. DLB is still lagging. And if you talk to clinicians and to PIs, they're all really doing, as Alexander just said, behavioral assessments to kind of set the stage for where the patient is, maybe an MRI to realize, oh, it's not vascular, it's not frontal temporal. So what are we doing? We're hoping to get at the third quarter our clinical data in a large phase 2 trial, and from there look at what are the parameters we can bring into phase 3. And one of the things that's concerning about this population, John talked elegantly about finding that right point in time to intervene. What happens with these patients in our observation is it's about 18 months. They go to 3 specialists. There's the movement disorders. They're talking to Parkinson's disease doctors. By the time they've gone to the 3 specialists and had on average 7 visits, a year and a half has gone by. And then maybe they've moved out of the window where they could be in an early trial and they're in a later trial. So I feel a sense of urgency around our progress in DLB just because we're not keeping pace with the patients. And since finally there's such great news about Alzheimer's disease, it feels like, well, my goodness, why can't we make more progress here? We will. We absolutely will. But it is clearly not where we are with AD, I think.
Got it. So as we, I know we got stuck in the topic of biomarkers, but as we kind of step back and think about the amyloid approach, broadly speaking, it may be helpful, Sumant, I think, just to understand as an investor, where else do you put your money? If you believe in the hypothesis and you still think there is room to optimize these molecules, whether it's a different species that you're targeting, or you're looking at BBB-enabled approaches, gene therapy, or even small molecules came in. So where do you really think about investment in this space going forward? It may be good to hear from the panelists, where else is the runway still left in this space of biology?
Most of the time, from an investor perspective, given the high risk, high reward nature of this business, I think it's all of the above. You really have to diversify. You can't just bet on one horse, for lack of a better term. So I think all the approaches that you mentioned will see some investments. People might pick some favorites there. But I don't think any investor will look at this as a one and done sort of thing. It's got to be a holistic approach. Many approaches at the same time will find favor.
Yeah, great. So I don't know if the others would like to add to that. Like, where is the next phase of innovation going to go in this space? I don't know. You touched on combinations as well, but I don't know whether who'd like. Go ahead, Todd, if you got ideas.
Happy to jump in here. As we think about the different things that we're talking about, we talked about staging, we talked about the biomarkers. To me, where it's exciting is where this all comes together, right? So if we think about the future and we think about precision medicine, but medicine overall, and we think about, okay, we're going to have all these different targets. To Sumant's point, we need to be funding therapeutics at all different targets because each one of them may be helpful at a different stage, in different combinations, for different targets, at different pathways, downstream, upstream. So we think about bringing all these things together, and I think that's where we will and we need to go, but it takes time. And that's where, if we look back at what has been done in cancer, I mean, that's, where they went. That's when we got a lot more effective cancer therapeutics, a lot more effective combinations. And then we just did a workshop on combinatorial approaches through our AMP-AD and our TREAT-AD and Model AD platforms. We're looking at all these different things. So we're getting there, but it takes time.
Anybody else would like to add?
Yeah, I would add one comment to that. I saw a little thing coming out here on the plane about when you look at the on autopsy, AD, about 10% of those patients have pure AD in terms of the protein, which leaves 90% for things that are interesting for Sumant to talk to investors about, right? There's room for every approach, whether it's inflammation or glial activation or whatever. I don't think there's ever going to be a silver bullet here that says that's the target. And so to your point, what's ahead, Murali? I think it is really going to be about the combinations of these various approaches, because the autopsies of patients tell us that, in fact, is what has been going on.
Go ahead, Jenny. Do you want to wrap up this section? Please.
Yeah, so I mean, absolutely think that the A beta antibodies are extremely important, but they're the beginning, not the end. I mean, it is fantastic to have, signs of success in a field because it attracts a lot of investment and also people having something to build on to kind of understand the biology and so on and so on. So certainly our portfolio is not just amyloid antibodies. So we have, one of the targets you've got there. I'm anticipating you.
It's a good transition to the next next areas to build on.
We talk about tau. Tau is another area that many companies are working on. We have some data reading out, hopefully at the end of the year, on OGA. So I think that all these other targets, we'll begin to understand how things work together. And also, a lot of both the misfolded proteins and other mechanisms cross across various types of neurodegeneration. So, an advance in one specific indication can quite often lead quite quickly to advances in others. So that's also, it kind of builds a bit like oncology. So I really think finally the decade of the brain.
That's right, slowly building up. And so speaking of building up, building up tau and propagation of tau is becoming an interesting area. It's taking center stage as well. But if one looks at the beta space and the tau space, the progress in the tau space has lagged behind, obviously, and still there's work to be done there. And as captured on this slide, I think there are different approaches being taken. There is, of course, the antibody approach. There are small molecules being taken. There is aggregation, or I would probably say more on the degradation side, efforts going on in that space. And even more recently, work going on in the ASO and siRNA type space. So multiple ways folks are attempting to modulate tau in a way that potentially could have clinical benefit. But I would certainly open up here again to the panelists here to kind of weigh in their thoughts on the different approaches that are being taken. Some of the highlights, for example, the studies with the ASOs, with the Biogen and ASO, looking to be providing some interesting clues here. I don't know whether, Jenny, you want to build on that a little bit. I know you touched on it briefly. You want to elaborate anything more before we move on to John for his comments? If he has any thoughts too, go ahead.
So sorry, I missed the very last point.
The different modalities.
I
think it's more on the different modalities.
No, clearly we're modality agnostic and have made quite a few recent investments in RNA and gene therapy, and there are lots of different ways of approaching these. And if we really need the best solution that addresses the patient need, and obviously that's going to be different for different. And also we could, but certainly the Biogen tau data was quite exciting, I think, for the field to see. And there's lots of, we saw earlier on in talks and the panel previously, there's lots of new data coming this year, and it crosses across all the different modalities. I think one place that for us we've seen a little bit less is kind of new approaches to amyloid, because now we've got the amyloid antibodies, and quite a lot of effort and work there, but are there sort of next generation amyloid therapies that we could be thinking about as a field? That's an interesting space.
Yeah, it's interesting in the tau space, the modalities, at least they're sort of going in parallel to some extent. But as you said, in the amyloid space, the antibodies sort of pushed ahead. The others are sort of catching up slowly in a way. John, I know this is an area that you and I, we've spoken about tau and proteostasis broadly. How are you thinking about this space with respect to druggability and the probability of this getting to the finish line in a meaningful way?
So I think that part of this is that, I'll go back to the multiple mechanisms, et cetera. Certainly, you can think about them separately, but when we talk about tau and neuroinflammation and perhaps even the synapse and synaptic dysfunction, is clearly, it's one topic actually. And I think and tau and neuroinflammation in particular, it goes back and forth. There's a nice review in this past fall in Journal of Neuroinflammation, where neuroinflammation clearly drives a lot of the tau pathology, and it's one of the ways to target tau is by targeting neuroinflammation. And there are some arguments vice versa as well, that the aggregated tau is actually inducing the neuroinflammation. I think the same story on synaptic dysfunction. And that's where the comment on proteostasis is really, it's the neuroinflammation induces proteostasis, which leads to the tau aggregation. So we can't, while it's multiple mechanisms, we really shouldn't be splitting it in that discretely. And then the question remains on tau, which I will say that I think even in the last year, we've made some progress on this, is that how does, like when we're targeting tau, what are we targeting? What's the downstream effect that we want? And I think there has been progress on that, and it's coming towards around trafficking of various proteins, axonal transport, trafficking of neurotransmitters within the synapse, which I think will, as we get better on that, we will make it will help define what it is, again, when we're targeting tau, what we're trying to do. And we are getting closer on that. On the neuroinflammation comment, there's actually out of the MGH group, they had a couple of presentations in, which I think, again, it goes towards how do you target tau? At AIC and at ADPD, that in their 3D model defining using a agnostic, just do whole scale proteomic targeting, and the targets come out in neuroinflammation. Actually, turns out P38 alpha was their number one target. But I think it's because of that it's, if you're going to target tau pathology, it's really targeting how neuroinflammation targets tau.
So maybe a follow-up. So, are the tau antibodies a long shot?
Pardon me?
Are the tau antibodies a long shot here?
I think it's the tough part of the tau antibodies is that inherently is putting you into the disease progression. Inherently, it's progressing over time, and it's just getting the readout is challenging. And for dose finding, proof of principle, I think that the tau antibodies fundamentally, personally, I think would work better in the tauopathies, where there is more triggering of actual a negative biological effect that you may be able to measure more directly early on. And again, coming back to some level of either clinical or biomarker improvement that you can see in phase 2b. So if they're going to be successful, it's likely in the context where there's an underlying mutant tau.
Alex, do you want to add anything to the tau conversation?
Maybe just putting the focus a little bit away from the antibodies to the other modalities. If you look at what Denali is doing, they are coupling their blood-brain barrier crossing tool with antisense oligonucleotides, and they have Alzheimer and also Parkinson programs there. I think that could offer a new, let's say, new modality pathway for those diseases. ASOs, in principle, you could also inject them intrathecally because there the effective half-life is something between 3 months and up to 6 months. Intrathecal injection is, of course, not the preferred route of administration, but if it would really require only 3 or 2 injections per year, it might be an option. But hopefully, Denali is going to be successful, and then you could give ASOs or siRNAs or whatever. You could then give them systemically. Let's see. I think it's an exciting field. We have the first ASO data now. Let's see what happens.
Murali, if I could, in our prep panel, we talked about the work of Jeff Cummings. He has this big wheel that shows all the different mechanisms. I think there are 140 trials just in Alzheimer's. And to your point, Alexander, I think 60 of them, 40 of them are orals. So who knows what's in there, right? And the rest are various antibody approaches. So the field is changing dramatically. I have a lot of hope that some of those things you talked about will come to pass in an oral, maybe not an ASO, maybe not intrathecal. At the end of the day, we have to pay for all this stuff, right? 6 million people, assume 2/3 of them get these medicines, right? So simpler administration is probably going to help us all in the long run.
Got it. Understood. So Todd, maybe one question before we wrap up. I guess the tau as well as the beta topic. As you think about funding and biomedical innovation from the NIA perspective, where are you going to start putting your dollars into going forward?
Yeah, so the comment that Lisa just made in terms of the cost, we think about not just the cost of the medicines themselves, but then also the cost of not having the medicine. So I think thinking about both parts of it is absolutely critical, right? When we think about it, I think you have to go across that landscape. We don't know yet, right? It's great that we have 140 trials, a lot more than we had 5 years ago, right? But how many trials are going on right now in cancer? Just saying, I think we have work to do. And I think that, we don't have a silver bullet. I think the comment that Jenny made, amyloid, it is a start. It's exactly right. And so when we fund, we really I think we funded over 40 targets over the last few years just in our small business programs across AMP-AD and those programs even more. So really looking at it from a holistic picture. The other thing that we haven't mentioned yet, and especially when we think about combinations down the road, is the non-pharmacological interventions, which, Alexander, I think, mentioned the behavioral side, and that's where the non-pharmacological interventions could be really helpful as well. So thinking about it all together, it's just, we can't put all the money in one basket, in my opinion, and that's how we're looking at it at NIA.
Great. It's great to have some of this section, right? I know the third bucket we want to touch briefly is on neuroinflammation, and I know I think John touched on it earlier on. Certainly a lot of activities going on, and we sort of lumped in neuroinflammation, and I guess the other word was neurometabolism together. And it's a lot of activity going on in different pockets, and there's a lot of potential here as well, but it'll be interesting to hear the panel's thoughts on that. Let Sumant lead that off.
Yeah, sure, thanks. So this is a bit of an alphabet soup here with the neuroinflammation side, but I'll start with the metabolism side because investors have been able to hide behind the weight loss theme last year. Clearly, Novo is running a trial with their product in Alzheimer's. I'm going to ask a question of Jenny here. What's the temptation?
Where is your trial? When are you starting a trial? That was a question,
yeah,
right?
Kind of paraphrase that, but I was going to ask, what's the temptation within Lilly to do that?
I would say the thing is we don't have to wait for Lilly to do this. Others have already done it effectively. There have been trials of things that affect metabolism in Alzheimer's. There's an ongoing trial in Parkinson's.
Right.
It's Novo. So I think, obviously data will emerge. You also get data even without trying as a drug, as it were, is launched into the market, or different mechanisms are being used by increasing numbers of patients in the population, you automatically will get data from that. So, we're going to find out but it is, clearly when you have a whole new area of interest, the obesity field has exploded in the last couple of years, at least externally. There's an interest to trying these things for everything, as the previous, not Lilly, I'm just saying the world immediately comes up with all sorts of suggestions for practically everything. But that doesn't necessarily mean that all of them are the most appropriate use of your dollars. You have to figure it out.
Then just going on to the neuroinflammation side, and I know that Todd and I have to kind of hedge our bets. We look at everything. But if I had to put you all on the spot, neuroinflammation wise, what do you think would be the most interesting? Is it TREM2? Is it something else? So we'll start with Lisa and then move down this way.
I thought he was asking. No. Oh, I'm probably the wrong person to ask Sumant. I will let some of my PhD friends and colleagues address that. There has been a fair amount of work in TREM2 and middling progress, but let others address that, please.
So move on to John then on that.
I think so. I'm not sure that Novo's concept of GLP-1 targeting is as a metabolism. It's the work on GLP-1. There is an anti-inflammatory effect, and I believe that primarily their rationale and this is what their Philip Shelton has presented, their PI. Is towards actually blocking effects of neuroinflammation. GLP-1 has clearly some anti-inflammatory effects. Again, it's anti-inflammatory not in the sense of reducing inflammation. So it's not targeting microglia astrocytes, but it's really targeting the innate immune pathways inside the neuron. And that's in the animal work, it does that actually quite well. And in that respect, the phase 3 trial of semaglutide may actually end up being singularly the concept of neuroinflammation being tested.
Yeah.
In my mind, and then we'll see.
Yeah.
I think even their view is that it's a good set of arguments to do it, but we'll find out.
Yeah, I mean, this is a space where there has to be significant amount of work to be done, because you can pick your favorite target, right? You can pick TREM2, you can put GLP-1, you can pick RIP kinase. Everybody can make up their own ideas around why that would be an interesting target, but I think it's an important thing for the field to think about how do we really test this effectively in the clinic, rather than going one after the other and hope you're lucky and successful. Don't know whether anybody would like to wrap up this section before we move. I think we got about 10 minutes, maybe.
If I may just make a comment about the neuroinflammation hype that has been going on, or is still there. Well, I do think that Alzheimer's primarily is not a neuroinflammatory disease. It's a neurodegenerative disease. Neurons are dying. And that, of course, then might cause an inflammatory process. We did some experiments where we could see that, yes, there was a neuroinflammatory component. But when we were able to remove the amyloid, the neuroinflammation was gone without any treatment. So I think, is there space for a neuroinflammatory treatment? Yes, right now there might be. However, if we would have a treatment that would remove at least toxic species of amyloid, there might be no need for any neuroinflammatory treatment anymore. Now, will we have that very, very effective anti-amyloid treatment? I don't know. But if we would have, I don't think, and then it doesn't really matter which receptor, which neuroinflammatory pathway would there be. If there is no amyloid toxicity, there will be no neuroinflammation, then there won't be any need for an anti-inflammatory treatment. That does not mean that for the next decade or so we might not need that, because we might not have that super effective anti-amyloid treatment. But I think I would still focus much more on the neurodegeneration than on the inflammatory part.
Great. And that sort of brings up to the next topic here very briefly, which is Alex pointed out, if all the toxicities can be addressed, at the end of the day, it comes to synapse, right? Can you get your synapse to work the way it's supposed to work? I'm not using the word regeneration of the synapse here, but that's going to be a topic for a different day. But there are a number of approaches that are being tried, and many of you are part of this experiment here, looking at different mechanisms to protect the synapse, whether you call it synaptic genesis, or you call it synaptic strength improvement, whatever terminology you want to use. At the end of the day, these are mechanisms that can, have effect rapidly, maybe in a symptomatic manner, but also may have potential for disease-modifying approaches. I know Lisa and the team is working on a couple of those ideas. Others are thinking about it. But, how is the world of that symptomatic and synapse protecting mechanisms playing out? Because I know there are great experiments being done, but again, getting an answer early on is challenging in this space. Biomarkers are going to be certainly helpful, but I would like to maybe hear about general thoughts around synaptic protective mechanisms, if that can be shared. Who wants to go first? Jenny and Lisa both are raising their hands.
Yeah, so
Go ahead.
I mean, I think even though sort of donanemab and the other molecules in our portfolio that are, the amyloid antibodies are, of course, aiming at being disease modifying, certainly we don't think there won't be a need for symptomatics, because, as you say, unless you actually achieve which would be fantastic, prevention, and we have some efforts going towards seeing if we can prevent the onset by treating people much earlier. But assuming that we're not going to be 100% successful at the beginning, then clearly symptomatic therapies are really important. And actually, we have quite a lot of effort ongoing now to look at this last word on your slide, on your first line there, of resilience, because it's clear that different patients, or across a patient population, even with apparently the same level of amyloid, the speed of progression and the kind of deficits that people demonstrate are different. And so if we cannot try and identify what is it that makes some brains more resilient than others, then maybe that gives us some clues as to how we can improve one of my colleagues referred to this as we've put the fire out in the kitchen now, so how are we going to remodel it all? So, and that's an interesting area where, again, certainly speaking as a kind of we're obviously looking to collaborate with small biotechs. We'd be really keen to see a little bit more investment in this resilience space and rebuilding, or not necessarily rebuilding, but protecting the brain.
Go ahead, Lisa. You had a comment to add to that? Yep.
Thank you, Jenny and Murali. So we had an interesting discussion. Somebody mentioned Philip Shelton, so he's a PI, and one of the things he was talking about is, look, you can target amyloid, you can target tau, and many companies and approaches do. He goes, what actually happens, what leads to the loss of memory and loss of function is the damage to the synapses. You've talked about this quite a bit. Synaptic function, the connectivity between the synapses, it's what happens first. And so coming over this morning, I was thinking it's sort of like an income statement in a balance sheet, right? Neurogranin might give you, it's like a balance sheet. It's a picture at a point in time. The synapses are up, they're down, whatever. Whereas what we looked at recently was EEG data showing the function of the synapses real time. We were so excited about this data because what you could see was the synapses having been retracted. After 4 weeks of dosing, you saw less theta band wave. Theta are those loopy, slow waves, which you look at and you say, oh, I bet that's what happens when you get dementia, right? The alpha waves were much better. Most importantly, it's the connectivity, how all those regions of your brain work together. You saw a directional change towards a reversal, towards normalization. And the key thing was, this was a 4-week study. I always say to investors, all of us sitting here have some level, sadly, of amyloid in our brains. It's been here for decades. Isn't that what we know? And so in 4 weeks, to see the return towards normal of synaptic health in these patients. So it was exciting to me, both for our data, our drug, but also to look at EEG as a tool, which is inaccessible. It is accessible, inexpensive. Patients can get to it. It's not a caregiver burden. So there were so many things about our recent look at the protection of synapses that was positive, both our ability to affect it and why it can be important to the field. And others are working in this area too. We also looked at SV2A in another study, and that had less of an immediate impact. But this recent work was very positive as far as we were concerned.
Really exciting space where we start hearing all of the pieces coming together at some point. And we wrap up the next couple of minutes.
If I could just add to that, Murali, I think it goes to a point that the field needs to embrace a little bit more, that a disease-modifying drug can have a symptomatic effect. Mm-hmm. Yep. And that's not actually, doesn't mean that it's not disease-modifying. And it's and it's only simply a matter of, are you in the stage of patients who can show that symptomatic effect? And that comes from, in other contexts, we don't make the equation that if you have a symptomatic effect, it's not disease modifying. No. In rheumatoid arthritis, the anti-TNFs are disease-modifying drugs. They have profound symptomatic effects. Yep. In cystic fibrosis, where I worked on previously, it's clearly a disease-modifying drug, but the first evidence of efficacy comes from there's an improvement in FEV1s within 2 weeks even. So it is this dichotomy we need to get away from. Mm-hmm. And a good disease-modifying drug, if it's in a patient that's early enough, should in fact have a symptomatic effect.
Yeah, that's a good point. Go ahead, Sumant. Let's spend the next few minutes wrapping it up. I think we are almost coming to the top of the hour.
Yeah, so
getting to the bottom of
the point I'd like to make here is that, we heard Harry talk about the antibodies, the approval of lecanemab, and one could argue about the efficacy of those kinds of products. But what is inarguably true, I think, at least from an investor perspective, is that investors now know that the FDA is open to approving such approaches. So that opens up the floodgates. It's a bit of a rising tide. With that, I'll actually ask a question of my co-chair here. It's a bit of a provocative question. What made AbbVie go into make a big splash in neuropsychiatry versus Alzheimer's, for example?
Is that a question for me?
Yeah.
Well, there's a lot of opportunity, as you very much understand. And again, in the context of this panel, I think we talked quite a bit about Alzheimer's disease, but there's a clear overlap with psychiatry. So if you think about psychosis in Alzheimer's, agitation in Alzheimer's, these are all neuropsychiatric illnesses, for example, which again is really important to the disease burden in Alzheimer's or Parkinson's. You deal with psychosis in both indications. So, a place like psychiatry, the breadth of areas that you can get into, which overlap between both psychiatry as well as neurodegeneration, is quite vast and varied. And I think when we thought about making investments in that space, we look at that breadth, how much we could get into, not just locking into a particular disease state and say we're only in Alzheimer's, but look at broadly as a neurodegeneration play and the overlap with neuropsychiatry, for example.
Any other last minute perspectives on where things are headed, Todd?
Yeah, I'm glad that Lisa brought up the EEG, because at the clinical stage, we do have a lot of great tools. We mentioned the plasma biomarkers. EEG is definitely one of them. Other technologies are coming on board. The one thing we haven't discussed, however, is the preclinical stage. And as we think about all these different potential modalities and we talk about eventually doing combinations, we're going to need insights prior to the clinic. And so, thinking about how can we do this at the preclinical stage, we've invested a lot at the NIA in terms of different models, whether they be early or they be late onset models, thinking about microphysiological systems where you may be able to get a readout on something like neuroinflammation. So, really important to think about that as well. And I hope that the confluence of technologies will have an effect both preclinically and clinically in our ability to test all of this out.
Thank you. Great. I'd like to thank the panelists. I'm sure there's plenty to talk about, but really great panel, and thanks for listening today.
Thank you. Sorry?
In the olden days, we called it animal pharmacology.